I am Yejin Kong from South Korea attending Seoul Foreign School. This poster is on the topic of a novel shuttle of therapeutic method against Huntington's disease. The main guiding question this study seeks to answer is how does the insertion of potentially protective microRNA types encapsulated by isolated exosomes positively or negatively impact brain cells dying from Huntington's disease? Huntington's disease is caused by a mutation in the Huntington gene, where the CAG sequence is overly repeated. Over the course of a lifetime, the impacts of this mutation are exaggerated and eventually cause more and more brain cells to die. While the link between microRNAs and this disease has been established, this study explores how that relationship can find a cure. To simulate HD conditions, 6-hydroxydopamine, or 6-OHDA, was used on A172 glioblastoma cells and treated with microRNAs encapsulated by exosomes, as they can't move on their own. The types of microRNAs used were also strategically selected based on pre-existing research and studies. Finally, this technique also has the possibility of being used for other diseases that are affected by microRNAs. The goal of this experiment was to help revert an HD-damaged brain into a healthy one, as shown in the image above. To differentiate the A172 glioblastoma cell into a neuron cell, it was treated with retinoic acid. Then, the total exosome isolation kit was used to isolate exosomes from the cell. The differentiated A172 cells were treated with 6-OHDA to decrease cell proliferation and replicate HD. Finally, the microRNA types shown in the table were inserted and observed to find the cell viability over 96 hours. On the graph, we can see how effective each microRNA type was in protecting the neuron cells. More information can be found in the link attached to the upper right corner of the poster, but the main findings shows that all three types were successful in increasing cell proliferation, indicating that the proposed therapeutic method was effective on protecting brain neuron cells. To conclude, I'd like to discuss the potential extensions this initial study can inspire. First, there are dozens of more microRNA types that showed a relationship with HD, and only the ones with the most dramatic differences were chosen. While there would have to be a process of elimination for efficiency and feasibility, there's still a more than likely chance that there's at least one other microRNA type that shows drastic increases in cell proliferation. Next, while the study showed a positive relationship, the impact of the amount of microRNAs wasn't studied. Since the focus here was to see if the microRNA insertion was beneficial, specifics such as the amount wasn't explored, and this variable has the potential to change the results. Lastly, another variable that wasn't explored was time. This experiment only observed the samples for 96 hours after treatment. If this technique is used on patients, they would have to manage it for the rest of their lives. So, observing the samples for at least a few more months could present valuable information. Finally, we have the references in the lower right corner. Thank you for listening to my presentation.